Good evening, everyone. It's 8 o'clock on my watch, so I think we will go ahead and get started. I'm Dr. Pitsukiwana. I'm not Chris Matthews. I'm just using his logins to help host the session. And I'll just quickly share the guidelines for this session. Dr. Richard Friedland has joined us on this call, and he'll do the introductions for the wonderful speakers who'll be sharing with us today. Just as a couple of guidelines, we will do the presentations, and then at the end of the presentations, we will have time for questions and answers. So if you have any questions, please feel free to either put them in the chat or you can raise your hand, and I'll just make a note of your hand. And then once we get to the question and answer session of the presentation, we'll then go through the questions. But for now, I'm going to hand over to Dr. Richard Friedland to introduce the speakers for tonight. Thank you, Richard.
So Pitsy, thank you so very, very much for that introduction. And colleagues and ladies and gentlemen, what an enormous privilege it is to be hosting the first of 7 series in what is probably the most exciting neuroscience development globally, and to be able to host 2 of the preeminent experts in neuroscience and neurosurgery globally, one in South Africa, from South Africa, and Professor Mike Sugru, all the way from Australia in Sydney. And thank you for joining us, Professor Shiger. It's 4:30 in the morning in Sydney, and we appreciate the sacrifice you've made. You know, this field of neuroscience and the ability now to be able to understand brain mapping and have a GPS coordinates, as it were, is revolutionizing our understanding of the brain and the application and potential treatment modalities. And it's a complex topic that Christos Profius and Mike Sugru will hopefully be taking us through step by step and demonstrating these extraordinary advancements. I just want to be able to read out the CVs and background of both of our speakers, just for us all to understand and appreciate the enormous expertise of our guest speakers and the expertise they are bringing to bear in South Africa to the benefit of many, many of our patients. Beginning with Professor Mike Sugru. He's one of the leading neurosurgeons and researchers in connectonomics, and I think he'll explain what connectonomics is, but he's lectured around the world on the management of glioma and the use of connectonomics, and having performed over 3,000 brain tumor surgeries, and also published over 250 peer-reviewed articles. Quite extraordinary. He's currently a neurosurgeon at the Prince of Wales Private Hospital and serves as the founder and chief medical officer of Omniscient Neurotechnology. This is an Australian startup with a mission to really develop and move neurosciences beyond evidence-based medicine and onto data-driven medicine with a revolutionary machine learning technologies. And Mike, Professor Sugru, will be following our first speaker, who is Dr. Christos Profius. He's got an extraordinary CV. He started studying, did his MA at Cambridge, and then went on to study medicine at the University of Oxford. He qualified in an MA at Cambridge with a double first class, and then completed his medical studies at Oxford. He's completed his neurosurgical training at the Wits University and achieved his fellowship, a neurosurgery fellowship from the College of Medicine in South Africa. He has also recently completed the Charles Teo Minimally Invasive Neurosurgical Fellowship under Professor Charles Teo at the Center for Minimally Invasive Neurosurgery in Sydney, Australia. And it was really during this time that he undertook advanced training in brain mapping that Chris was going to take us through tonight, and what that means. These very techniques under Professor Mike Sugru. And as we know, Professor Sugru is a leading expert in this. Christos is a super specialist in brain tumor and vascular neurosurgery. He's got a special application in the special interest in the application of keyhole surgery and endoscopic techniques to vascular, skull-based, pituitary, and neuro-oncological procedures. He is also the head of department at the neurosurgical unit at Helen Joseph Hospital in Johannesburg, under the auspices of the University of Witwatersrand. And we're privileged to have Christos working at Netcare Moore Park Hospital, as well as Netcare Linksfield Hospital. So I think you can see, ladies and gentlemen and colleagues, what an extraordinary privilege it is to have these two exceptionally well-trained, erudite professionals take us through this incredibly exciting topic and new development in neuroscience. And without much further ado, I'm going to hand over to you, Christos. I will say just one thing. I have referred several patients to Christos for TMS, and amongst many of them, I have seen the most remarkable improvement, particularly those with refractory depression and other mental or psychiatric illnesses. Over to you, Christos.
Thank you, Richard. Thank you for the kind words. I'm going to get straight into it. Thank you for your time, everyone. So the human brain truly is the crown jewel of human creation. It's why we walk, it's why we talk, it's why we have thoughts, it's why we have plans. Ultimately, it's what makes us human beings. Understanding what is at its core is almost a question that's old as what is life itself. In order to understand our current developments and the current way we can think of the brain with new technologies such as machine learning, we do have to go into the past a little bit. So how does the brain work? Well, Aristotle first theorized in 300 plus BC that it was a radiator to the heart.
Presumably the moral imperative to bring freedom and democracy to Russia, Alexei Navalny,
then Galen took on a role where he started to think that perhaps this organ in the brain, and particularly the ventricles, are the seat of complex thought. But for over 1,000 years, there weren't many developments in terms of understanding how the brain worked. Vesalius in the 1500s made very detailed anatomical sketches. And then people started to look at the structure in a lot more detail. In terms of how it worked, Galvani came up with the concept that electricity is the currency of the nerves, which sort of brought us a bit closer to our current understanding. However, if we think about how we think about the brain today, the 1800s had a huge influence. The story of Phineas Gage, a railway worker who had a steel pole go through his frontal lobe, and then he became very impulsive, very rude, very difficult to deal with. And that sort of gave people the idea that perhaps the frontal lobe has some role in these functions. Broca published a paper on 12 postmortem patients who had problems with speech and localized the speech area to what we know as the Broca area. And Wernicke localized the comprehension layer of the brain. But these important developments started leading to a thought process where all of us today think of the brain that certain parts do certain things. And if certain parts of the brain don't have an obvious function, then we tend to call them non-eloquent. The 1900s had some interesting developments. Kajal and Golgi started looking at the brain under a microscope and mapping neurons in exquisite details. But the ultimate brain map came from Corbinian Brodmann in 1909. So he put together a brain map of roughly 47 brain areas, which we are all taught in our specialities in relation to neurosurgery, to neurology, to psychiatry. And we almost taught that it's the ultimate map. Penfield modified this with the Montreal procedure and he gave rise to the thought process of a cortical homunculus. But ultimately, if we look at the current understanding and clinical practice of the brain, the Brodmann map is the most detailed view that we have, where it's got obvious areas that serve important parts, such as the motor cortex or the sensory cortex or Broca's and Wernicke's, as we mentioned. But does it really describe how the brain works? And ultimately, when we look at this map, is it the map that we think it is, where we have exquisite detailed, or is it somewhat fuzzy? And should we really be looking at ways to improve our understanding of the brain? In order to proceed and understand how we can understand the brain today, we need to look at a concept called the connectome, which for the past 20 years has led to some astonishing developments in neuroscience, but unfortunately as clinicians, we're very rarely exposed to. So in simple terms, what's the connectomes? Well, ultimately, it's a matrix that represents all the possible anatomical connections between the brain's elements, which, depending on the scale, can be neurons, or it can be lobes. Now, when we describe the connectome, we can look at it in terms of space, and we can talk about a microscopic connectome, where we're looking at individual cells, or we can go up to a macroscopic connectome, where we're looking at centimeters of cortex. There's also very complex time variations. So if you're going to start looking at changes in the connectome, the time factor is important, and that can be down to a few seconds when you're looking at fMRI, or it can be a few milliseconds when you're looking at electrode neurophysiology. But ultimately, we're studying the human connectome. And one of the big problems is it's incredibly complex. We have an estimated 86 billion brain cells with 1 trillion supporting cells. And on average, each one of these brain cells connects about 20,000 times to each other to make more than 100 billion connections. If you're looking at the action potentials in this organ, it's firing at about 17 trillion electrical signals per second. So ultimately, we've got to look at technologies where we can map the brain, which will give us an idea of how it works. If we were to be absolute, probably one of the best technologies to look at is an electron microscope, where we could take our time and map every single brain cell and see how it connects to other brain cells to get an ultimate idea of how the structure is connected. The big problem with that is the scale of the human brain. So White and Brenner in the 1990s decided to map the C. elegans, which has 302 brain cells and about 5,000 synapses. The problem is this project with multiple PhD students took more than 10 years. If we extrapolate, it takes about 800 man-hours to map 1 millimeter cube of human brain cortex. And the problem with that is when you upscale it to a mouse cortex, it takes a decade. When you upscale it to a human cortex, it would take us about 1,000 years at this rate to map every single connection of the brain. Not only is that not possible with current technology, but it also creates a data storage problem. If we're looking at the actual brain, if we wanted to store all that information, it would be equivalent to 1 zettabyte, which is roughly the global mobile phone traffic data that's used on an annual basis. So everyone's WhatsApp message, everyone's Instagram post, it wouldn't be enough to store that over a whole year. And if we compare it to the Human Genome Project, that was just 1.5 gigabytes of data. So clearly, the electron microscope is not the way to do it. A very interesting technology, which a lot of us as clinicians are familiar with, is the MRI, or more specifically, the fMRI. One, we can look at much larger areas of the brain and see how they connect to each other. So it makes data more usable and more computable. But the other interesting aspect of this is, from a transitional point of view, any decent hospital has an MRI machine. So if we could find interesting ways or very clever ways of mapping the brain with an MRI, it's potentially applicable to many different hospital settings and applicable for patients. So how does the brain work? If we explore this question currently, then one must mention the Human Connectome Project. The Human Connectome Project started in 2009 under the NIH in the US, and it was funded by approximately $40 million. And what happened was the leading institutes, brain imaging institutes of the world, i.e. Washington, Oxford, and Minnesota, were given a grant to make new brain mapping techniques and also get very detailed brain maps of patients. Ultimately, they mapped about 1,200 subjects aged between 22 and 35, i.e. young adults. And a lot of the patients were twins or related. A subset of patients also had very detailed behavioral testing. The project itself took 2 years of upgrading MRI hardware and coming up with new sequences before a patient was scanned. In fact, 3 Tesla MRIs were almost made specifically for the project, and Siemens at the time said it was just a once-off project, but eventually converted that into the 3 Tesla Prisma MRI. So about 1,100 patients were scanned under the 3 Tesla MRI, and each patient was scanned for an average of 4 hours, which any MRI technician will tell you is an excessive amount of time. Some of the patients were scanned at 7 Teslas. Some were scanned with something called a magnetoencephalogram, which looks at a very detailed structure, but is also able to get very good temporal resolution of scans. And ultimately, we had different types of brain sequences whose purpose was to provide an ultimate brain map. So if we're looking at the brain sequences that they used and what other neuroscientists are using to map the brain, the ones that are required is a structural brain scan, which is usually a T1 brain scan where we're just looking at the brain structure. Something called the diffusion brain scan, which I'll explain in some more detail very shortly. And that allows us to trace all the brain tracts. And then a functional MRI scan, where we're able to analyze the blood flow in the brain and by extrapolation, figure out which parts of the brain are active. So structural MRI is pretty straightforward. Most of us dealing with patients in neurology or neurosurgery or psychiatry are aware of what it is. However, I'm just going to go into a little bit more detail about the diffusion MRI. So the principle of a diffusion MRI is that water is present in the brain, and what it likes to do is have Brownian motion that diffuses randomly in any direction. However, when we have water molecules in brain axons, the axons have lipid membranes, and they limit the movement of water molecules up and down an axon, but not from side to side. So when we actually apply pulses to the brain, we can follow in which direction these water molecules move. Now, because they're restricted by axons, they have an anisotropic movement. So rather than being isotropic where they move randomly, they move up and down axons. And that gives us data which allows us to extrapolate where brain tracts are. More specifically, we're able to calculate things like eigenvector and eigenvalues and look at each voxel in the brain and determine what's the predominant direction of a water molecule and figure out where a brain tract is. Now, there's many different ways of doing this, such as DTI, which some people might be aware of, but more modern ways can use CSD or constrained spherical deconvolution, where we get a much better resolution of crossing brain tracts. But ultimately, what diffusion MRI allows us to do is get a very detailed maps of where brain tracts are. Now, the next MRI sequence of the 3 that we discussed that we need to know something about is a functional MRI scan. So what a functional MRI scan does is it uses something called bold signal to extrapolate where blood is flowing. So when a brain is active, more blood goes to an active part of the brain, and that carries within it hemoglobin. When hemoglobin loses oxygen, it's got magnetic properties. And by that, we can extrapolate where the blood is going. Now, traditional functional MRI is something called task-based functional MRI, where we're asking a patient to move an arm or talk, and then extrapolating which parts of the brain light up in relation to that movement. Now, very interestingly, a PhD student in 1995 in Milwaukee called Biswald was doing an fMRI study, and what he picked up was that patients that were waiting in the scanner, not doing much, had very predictable parts of the brain lighting up together. And this was across many different patients. And this gave rise to the concept of brain networks, i.e., when we're at rest, different parts of the brain fire together, and we can see this on functional MRI imaging. And in essence, because they're firing together, they're bringing a common function together, which makes a network. Now, in order to figure out which parts of the brain are connected, there's been very clever statistical methods devised. And one method that's used in a resting state functional MRI to figure out which parts of the brain are talking together as opposed to which parts are noise is something called ICA, or independent component analysis. And if we were going to give an analogy, it's able to look at the brain waves or brain signals. And if we made an analogy of multiple speakers speaking at once with a crowd, and we had some microphones that were picking up everyone's sound waves, what ICA is able to do is essentially break down those brain waves and tell us who's talking to who. And in the same way, this method allows us to know which parts of the brain are talking to each other. So ultimately, if we look at the success of the Human Connectome project, what it did do is it provided us with large amount of brain scans, which were publicly available, which allowed us to make very detailed brain maps. Those were able to be linked up with behavioral and genetic data. And essentially, it became an open source platform where neuroscientists from around the world were able to access these scans and perform very detailed studies. It's led to close to 1,000 publications in high caliber journals. And when we look at some of the publications and how they've changed the way we think about the brain, it's really monumental. And as clinicians, we've really been left behind in terms of the way we understand the brain. So I'm just going to talk about two very important publications which have really shaped the way we can see the brain. One of them comes from a radiology registrar called Matt Glasser, who in 2016 publicized the most detailed brain map. So initially I showed you the 47 area Brodmann map. Matt Glasser used machine learning where he made a classifier which involved looking at cortical brain thickness, myelination, functional MRI studies, and it also took into account previous brain maps. What he came out with was a brain map which had 360 areas on each hemisphere. So in total, 720 areas for the brain. If we compare this to Brodmann's map, so Brodmann had initially 47 areas, only 23 of those areas remained in this parcellation scheme, and only 10 of them remained in their original form. What this did is it actually paid tribute to the brain as a complex structure. And a lot of these areas don't follow gyri, or don't follow sulci. In fact, some of them are entirely in sulci and many of them cross gyri. So we think of the brain as lobes, but the function of the brain itself does not resemble the way we've constructed in our minds of how the brain works. The most important aspect of this is it's given us an alphabet, because what Glasser did then was he was able to put this machine learning classifier into brains that he hadn't studied, and the machine learning was able to locate these areas with a high degree of specificity. So when we're studying different people's brains, if we're talking about, for instance, an area 8C in patient X, we can find that exact same area in another patient Y. The next study highlights the importance of what machine learning can do in mapping the brain and how we can even use it to diagnose disease. So this study comes from Drysdale, from Cornell University, and they're a major psychiatric unit. And what they wanted to do is they had a lot of patients with resting state fMRI data, and they wanted to look at the connectivity in these patients of the limbic system and the frontostriatal network. Once they looked at that connectivity, they wanted to compare it to a normal group of patients. So they used machine learning to say, okay, we've got a whole group of patients, 300 plus, with major depression, and we've got 300 plus patients who are completely normal. Can you tell a difference between the connectivity of the limbic system and the frontostriatal networks between the two patients? Impressively, the machine learning could do exactly that. And what it actually came back with was that what we call depression is actually 4 different types of diseases, or otherwise known as 4 different biotypes. And a lot of the differences between the biotypes you can tell clinically, but not strictly. So some areas you can't actually tell the difference when you're looking at them clinically. More importantly, however, when they put their model together, Then they got other units in the country to send them blinded scans. So they sent them a mixture of normal scans and scans of patients with major depression. And when they put it through their machine learning classifiers, they could detect who had major depression at a sensitivity of about 90% and a specificity also of about 90%. And this was without knowing any prior history of the patient, never talking to the patient, never taking a history. So what this proves is that through very detailed brain mapping algorithms, you can actually pick up differences in people's brains when they've got a major mood disorder and when they don't. Furthermore, when they classified the different biotypes of depression, they could also tell treatment response of those biotypes. So a biotype 1 has a higher success rate to TMS as opposed to a biotype 2. So ultimately, how do we put all this together? So the Human Connectome project was a great project. It taught us a lot. The neuroscientists over the last 20 years have had major developments of the brain. The best way to describe this is to actually look at the cerebral networks in more detail. So what is a cerebral network? What a cerebral network is parts of the brain that are synchronously active and bring about a common function, i.e. they fire together at the same time to bring together a function, a common function. And what it does is it debunks the theory of eloquent versus non-eloquent brain. Just because there's parts of the brain that we can't describe in as much detail as the motor cortex, it doesn't mean that they're not doing something important. There's many subnetworks, but what I'm going to do is I'm going to focus on 7 main brain networks, which bring together a lot of the common brain functions. Some of these networks you've heard about, maybe not thought about in this context, but you have heard about. Other ones you may not have heard about. So what I'm going to start off with is the 2, what I call input-output networks. So the first one is the sensory motor network, and that has 30 parcellations. So each one of those areas on the Glasser atlas that we talked about is called a parcellation. And generally, the areas that the sensory motor network involves is the motor cortex, the sensory cortex, and the auditory cortex. So when we study these areas in resting state functional MRI, i.e. when patients are at rest, these areas fire together. So we don't get firing of the motor cortex independently of the sensory cortex. And it sort of makes sense because in order to move an arm, you sort of need to know where it is in space. The visual network is responsible for sight and visual processing. And you won't be surprised to know that it's the largest network with 52 parcellations. And that makes sense given that we visual. So human beings rely on vision as the main sense. The next network is the limbic network, or a primal or ancient network. And next week, when we talk about brain evolution and architectonics, and when you start looking at how the brain's evolved from the lamprey, it sort of makes sense where its position is. And its position sitting in the medial temporal lobes, the inner frontal lobes, and the amygdala. So all the areas that we've learned and we know about, and they're responsible for emotion, motivation, fear, and memory. Up until now, more or less, we've heard about these networks, even though maybe we haven't thought about them as networks. Now, what haven't we probably heard about? So the cognitive networks, the whole area in the cognitive networks exploded after Biswald in 1995 first discovered the Fmri. And one of the first networks to be described by Gracious was the deep mode network. So the deep mode network involves the medial frontal lobes, the cingulate gyrus around the precuneus, and the interparietal sulci. And this network is important for internal thinking, imagination, when we sit and contemplate our lives and how it relates to the universe. A competing network to this is the central executive network, which was basically defined by Swilly. And this one's responsible for working memory and external thinking. It's basically, we're using many of its elements now as we're focusing on this presentation to learn new knowledge and relate it to all our past working memory. And this one involves a lot of the lateral frontal lobes and interparietal sulci area. Now, the next network that I'll talk about is the Salience Network. And this network involves the anterior insular and the anterior cingulate cortex. And it's thought to have a role in switching between the deep mode network and the central executive network, i.e. internal thinking and external thinking. We can't really be doing both at the same time. So even if it's milliseconds apart, we tend to be in one network or the other, and the salience is thought to have this switching mechanism. It probably does a lot more than that. It was first discovered by studying frontotemporal dementia. And then there's a lot of work to suppose that it's involved in autonomic function in terms of if we fall in love and we have butterflies in our tummy, it's probably got something to do with that. But in a basic sense, it's important in terms of switching between a deep mode and a central executive network. The last network that I'll talk about is the dorsal attention network. We have a lot of visual information in our brains, and it's also a very large network with 49 parcellations. And it involves areas such as the frontal eye fields and interparietal sulci. And its importance essentially is to basically focus our brains on relevant information. So if you're following this presentation now, your dorsal attention network is telling you to look at the screen and maybe not at a painting or a couch or something like that, which is sitting behind you. So ultimately, why am I interested? Well, I'm interested because I'm a brain surgeon and I always had a passion about brain tumors. So when I finished my qualification, I wanted to learn a lot more about brain tumors. And I was fortunate enough to secure a fellowship with Professor Charlie Teo in Sydney. Professor Teo is one of the world's greatest brain tumor surgeons, does a lot of the difficult tumors that most other surgeons are not able to resect. And I was fortunate enough to do a fellowship under him and learn about his keyhole techniques and his approach to brain tumor resection. Now, in this time period, I was fortunate enough to meet Mike Sugru. So Mike Sugru was also a fellow of Charlie about 10 years ago. And he came back from Oklahoma where he had a glowing career and had resected more than 3,000 brain tumors and set up a project looking at very detailed brain maps and how they relate to brain function and brain tumors. He also introduced me to TMS. Now, when Mike got exposed to all the information of the Human Connectome project, which I described earlier, he wanted to know a lot more about it. And his quest started on finding out where these areas are on an actual brain, because Glasses Atlas published a virtual brain or an inflated brain. So what Mike did is he went on to study which parts of the brain these parcellations are located. He looked at all the diffusion imaging to find out how each parcellation connects to the rest of the brain. And he also looked at very detailed functional imaging to find out on functional imaging how each of these areas are connected and also describe their function. Well, when he put all this work together, Operative Neurosurgery in 2018 gave him the entire issue, and he published 18 papers on this subject with a very detailed interpretation of every single area. So ultimately, I finished my fellowship and I came back to South Africa, and I achieved my goal of learning how to take out brain tumors through keyholes and doing very large resections and thinking about how to make the operations as safe as possible. However, Mike Sugru introduced me to brain mapping, and that stuck with me because the first time I talked to Mike was on New Year's Day. And after about 15 minutes in the conversation, and after he was telling me about these networks and how they wired and what they do to the brain, and after he was sharing with me all the work that he was doing with TMS, I quickly realized that I didn't know enough about the brain. And I felt nervous to operate after that. So I had to retrain myself to think about the brain differently. When I was there, getting a brain map was very tedious. So it could take up to many hours, if not 12 hours plus. We had to use Linux programming. We had to convert imaging files. We had to use other software to image spots and figure out which one's noise and which one isn't. Thankfully, Mike took this task on, that we needed a much simpler way of analyzing brains, which made sense. And he went on to form Omniscient, which he'll tell us a lot more about later. Before I can go into how all this applies to patients, I do need to introduce TMS. So transcranial magnetic stimulation is a very interesting non-invasive stimulation technique. There's different types of TMS, but the one we're going to focus on is repetitive TMS, because essentially with the use of this type of TMS, you can actually modify the way the brain functions, and this lasts beyond the treatment period. Now, repetitive TMS uses magnetic coils, which are placed over the head, and they make eddy currents, and through the eddy currents, they can make electric fields. Repetitive TMS has two different types of protocols at its core. It's got continuous CTBS, which is CTBS, or intermittent TBS. The way all theta burst TMS works is that essentially each burst has 3 pulses, which are fired at about 50 hertz. And then there's an interval of about 0.2 seconds, and then another burst. With CTBS, this sequence is continuous and it lasts over about 40 seconds. With ITBS, we have a burst for 2 seconds, then a rest for 8 seconds, and then burst for 2 seconds to give a total of 600 pulses. The mechanism of this is thought to reflect long-term potentiation. And certainly when you're studying MEPs of the motor cortex, or how excitable the motor cortex is after treatment with CTBS, what you get is suppression of the cortex for about an hour. When you stimulate the motor cortex with ITBS, you make it more excitable for about 15 minutes. But the idea here is that we've got a sequence that can suppress the brain, which is CTBS, and we've got another sequence which can excite the brain, which is ITBS. So TMS was first being developed in 1985 and then evolved a lot up into 1990. And there's more than 100 clinical, randomized clinical trials in many different types of conditions, which have showed efficacy. In fact, for depression and PTSD, there is FDA approval for this treatment. But most of the TMS practice around the world is conventional. And what I mean by conventional is that a conventional target for depression is the left dorsolateral prefrontal cortex. And people use CAPS where they measure parts of the brain and try and figure out where these are. But as you'll see in the next hour or so as we're presenting, the DLPFC has 27 parcellations, and every human being has a different composition of these parcellations. So it probably doesn't do justice to the complexity of the problem. So what we can use then is brain augmented TMS. We can use very specific brain maps to figure out which parts of the brain are overactive or underactive, and I'll show you more information about this. Then we can map it with a navigation system when we're performing TMS. And we can also hit targets that are outside of the frontal lobe. And ultimately, we create a much more sophisticated type of TMS, which is targeting the exact problems based on personalized brain mapping. So in order to do this, when I got back, I realized that TMS wasn't being practiced in South Africa, or there was very little of it being practiced, and definitely not with brain maps in mind. So we got together and formed Connectomics, and its main mission is actually an education program. So what we want to do is we want to educate clinicians to think about the brain in terms of a detailed brain map, parcellations, figuring out which parts are active, which parts are underactive. And we also want to provide an open TMS clinic, which has at its core a navigated program. So once we make a detailed brain map, we've set up a TMS clinic where we can do very detailed brain mapping on the patients, and we can use navigation to map the exact parts of the brain that need stimulation. And we're obsessive about that target accuracy. In a nutshell, the way it's set up is that after clinicians are trained in brain maps, they might have a patient where they're interested to get a brain map. They can order this electronically, and then essentially the team at Connectomics will be informed that a brain map is requested. What Connectomics will then do is basically contact the patient, organize the MRI, and also upload the scan on the Omniscient platform and make sure it's working, and inform the clinician that the brain map is ready for viewing. Connectomics will also have a program where there'll be regular training sessions, and cases can be discussed in order to guide clinicians on how to read these brain maps and ultimately make TMS plans if they think that's appropriate. So once the clinician gets their brain map and analyzes it, should they think that TMS is appropriate, because that might just be for a diagnostic purpose, they can pick which targets they want to stimulate, and then that will be communicated across to Connectomics, and then the patients will be booked for their TMS. They'll have navigated TMS, and then essentially they'll get their treatments on the specific targets that were chosen. The patients will then go back to their treating in-charge clinician, and they'll have a report. And in the meantime, there's also a platform where patients are in the loop, where we're conducting different research projects, and we're able to ask them questions on different clinical scales, and also have a platform where we're able to give them information about TMS and have them complete consent. Okay, so I'm going to get straight into the brain maps, how they work. This is from the Omniscient platform. So when we're brain mapping, we're looking at a structural MRI brain scan. And what Omniscient does then is they use the diffusion imaging and overlay it on the brain map. And then they use machine learning to essentially map the 360 parcellations on each hemisphere from the Glasser Atlas. And this is a unique personalized brain map. These parcellations are a fingerprint for each patient. And Omniscient's figured out how to essentially map these areas to the brain. Now, what we're able to do then is we're able to select a deep mode network or a central executive network if we're looking at mood disorders, and we know exactly where it is. The interesting part comes next. What Omniscient has done, and Mike's going to give us a lot more details about this, is they've made it possible to for us to compare a patient's brain to a database of healthy young adult brains, which don't have a mood disorder or don't have any neurological problem. So in this patient, if we were interested in a mood disorder and we're looking at a central executive and a deep mode network, which I discussed earlier, we're setting up an adjacency matrix, which is comparing all the parcellations, so all those areas in the Glasser Atlas that belong to those networks. And what we're doing is we're comparing how connected they are to each other. What you'll notice is there's 4 colors. There's white, there's black, there's blue, and there's red. If 2 parcellations are connected by a white little box, it means that their connection is normal. So when we compare them to young, healthy adults, the connection between, the functional connection between those 2 parcellations is normal. When it's black, it means that there's too much variability between subjects to make a conclusion. When it's red, it means based on that functional imaging, that parcellation of PFM, for instance, and LD32 is overconnected. When it's blue, it's underconnected. And what we're able to do is look at columns. So for instance, in this patient with the mood disorder, we can see that this column here, which is a PGS and comes up fairly common in mood disorders, has 1, 2, 3, 4, 5, 6 abnormal connections. And what we can do then is we can locate exactly where that is on the brain and pick it as a potential target for TMS. So once you select on the adjacency matrix where the target is, you can see exactly where it belongs in the brain. So what we've done is we've figured out which part of that network is abnormal, and now we can make that into a personalized brain medicine target. So what I'll do is I'll just present some of the experience we've had with some of our patients at Connectomics, and then we'll pass on to Mike. So this patient here had a low-grade glioma in primarily their sensory cortex, but moving on into their motor cortex. And we can see this on the T2 imaging on the left. Now, this was a young adult, and we had a very frank conversation with her, and I told her the chances of this becoming a glioblastoma is very high, and that's essentially a death sentence. So we decided to have a very aggressive resection of this tumor. But the problem is postoperatively, as expected, and as she was consulted, she would have a weak leg and a leg that wasn't very good with proprioception. So what we did is we put it through the Omniscient platform, and we got the diffusion imaging, and you can see the hole where the resection was. And we figured out where the glass atlas was for this patient. And what we did next is then we started looking at the sensory motor system to pick up areas that were abnormal. If someone's normal and they don't have a problem in this area, this whole adjacency matrix is white. And the machine learning was able to pick up exactly where the problems in this person's sensory motor system were. And from that, we were able to pick targets, which we could hit with the TMS. So what we've actually got is before the treatment, she had a lot of problems in controlling that leg in space. And we can see she's trying to hit the pedal, but it's taking a lot of effort and it's very difficult. Well, by day 3 after the TMS, she was able to do this, where she regained control of that leg, was able to walk much better, and basically got the function of her leg almost back. Another case that I'm going to show here is a case of a patient that we didn't really think TMS was going to work. So it was a bit of a horrific work. It was a bit of a horrific story where there was a home invasion and the patient got stabbed in the neck, and he had an incomplete spinal cord injury, very weak in the limbs, some movement in the limbs, but not much. So we did do a brain map and we looked at different motor areas, and we found an area in the SMA that was overactive. So we combined that in a treatment plan together with the primary motor cortex and sensory areas. But what we got was a very motivated patient and an excellent rehab effort. But every time we were doing TMS, we noticed a difference. So this was him when we first started, and he had very little movement in the hands, just the fingers a little bit, not able to lift the arm, not able to extend it. So after the first day of TMS, he hadn't been on his feet, and we're able to actually get him off the wheelchair, and with some assistance from staff and family, we basically got him to start walking. So what we did is we did a pretty intensive TMS program with the rehab team, and every session that was passing, we were just starting to get more and more arm movement. Eventually he was able to lift the arm in air and get a lot more control. Walking started becoming independent, where he basically could push himself off the wheelchair and start walking. And then in the weeks that ensued, and with some more treatments and, very motivated rehab team and a very motivated patient, we got him on his legs to the point where now he's able to type on his computer and typing at a rate of 24 words a minute from what he's updated me. And he was even able to complete a 5-kilometer run. Now, the next case I'm going to show is a case where we had a brain bleed in the thalamus. So young girl had a previous bleed, stayed stable, and then after a few years got another bleed. Severe headaches, but very anxious and wanted it out. So we did some very detailed brain mapping and tried to figure out, which pathway would be the least traumatic to the major brain tracts, which we'll go into a lot more detail next week. And essentially the plan was made to resect it. And then we got the cavernoma out. But the problem post-op is, and as she was consulted, that she could suffer from fairly severe weakness. And what we did is we did the brain map, and we could see in the premotor cortex that there's an area 6D where essentially it was overactive. So we combined that in our TMS plan, and prior to the treatment, she was completely reliant on her husband. to basically hold her and give her balance and get her to walk. By day 3 of treatment, we got her walking independent. And then the last video was 2 weeks after the treatment, where essentially she was walking on uneven ground, much more independent. And the last we chatted and the last I saw a few weeks ago, her walking's essentially back to normal. Now, these cases sort of reflect some of the neuro rehab abilities, especially when you're using targeted brain areas based on very detailed brain maps. But we've also had very interesting results with our mood disorders. So this patient had severe anxiety, been to many specialists, frustrated by the fact that he had a very normal structural brain scan. However, when we were looking at his anxiety disorder and looking at his brain map and looking at his DMN and CEN, those brain networks for external thinking and internal thinking, we could very quickly see that even though his structural MRI, which we just showed is normal, his brain map is very abnormal. There's very many red and blue areas. And from this, we were able to pick a few targets, and not conventionally targets in the dorsolateral prefrontal cortex. So we did a target in the parietal area and the temporal area. And he had an excellent response to the TMS based on this process. So what I'm going to do, I'm just going to finish off with a video of him being interviewed, which was done recently. It's been 9 months since you had TMS for anxiety. Could you please describe your symptoms before you had TMS and your experience with anxiety?
Yeah, sure. I suppose the best way to describe it is your lives in fast forward with anxiety. Always, elevated heart rate, and because of that, I'm sweating a lot. Yeah, and just this constant feeling of anxiety. I suppose it's no better way of explaining it. I suppose, and it's always there. You battle to sleep, and your brain is basically in fast forward mode all the time.
And then it's been about 9 months since we've had the TMS. Have you noticed a difference since you had the TMS treatment?
Huge difference. I noticed the difference after the first day, that night. I noticed, well, I slept very well, first of all, and I don't have, my heart rate went down. It wasn't as elevated as before. And I wasn't sweating so much. After I think it was a third session that night, I suppose is the peak is in terms of how I felt, and I was feeling really good then. And ever since, it definitely made a difference to me. It's just I feel like I'm more in control with my thoughts. It's not everything is fast forward. It's normal pace. So it's made a huge difference.
Thanks, Francois. Thanks very much.
Okay.
Francois. So with that, I'm going to hand over to Mike Sugru, who I'm really excited that he's going to present, and I promise everyone you're going to be amazed with the work that he's doing. We're going to carry on next week with a presentation on cerebral evolution, development, graph theory, and the different white matter tracks before we go on to describe all the lobes of the brain, which is going to be followed on by weekly sessions. Thanks very much.
So, you've done a good job teaching someone something when they come and basically give half your presentation in this session before you. But I'll try to add on to what Chris has done. And what we're really going to talk about, and I think hopefully some of that speaks for itself. When we talk about transcranial magnetic stimulation, I've been in research and neuroscience and various things for 20-something years. And I've learned over the years that a lot of times you go to meetings and you see something that people present statistics or data or a trial or whatever, and you get very excited. And then you go back and you find out that, well, yeah, the trial shows that this is a 10% difference. You just have to trust the data. But it isn't that you walk in and immediately know that what you're doing is working. So for example, you know that prophylactics for DVTs works because you just, after a while, one day you think, I haven't seen as many DVTs. But it isn't that you go back and immediately notice that, hey, this is obviously works, and you just have to trust the data. TMS is not that. Okay, so TMS is one of those things that very quickly, after seeing lots and lots of neuroscience patients and the rates of recovery, you see the slope of that recovery change quite dramatically, and oftentimes in ways that are just no one in the building has a debate that this treatment is doing something to the brain. And one of the things that really surprised me, and one of the things that, I have no conflict of interest other than for TMS, I don't sell the device or anything like that. I just think that it's a tragedy that this treatment basically got pigeonholed in little parts of psychiatry and has been held back by kind of the failure to translate, given that in some patients this can have a really dramatic result. Now, what we know about TMS is that it rewires the brain. It changes plasticity. I'll even show you some data that or at least a very dramatic case where it actually even causes the brain to rewire in some cases. So when you think about this in a practical terms, given what we know about the science, given what we know about the numerous clinical trials, and given the obvious experience, what we really have to think about, what we're really doing with TMS or any other non-invasive brain stimulation, is rewiring the connectome. Rehab rewires the two. And when we do this together, we're basically editing somebody's brain connectome, their functional and structural brain connectome. Now, that always sounds like science fiction, and we always thought that what, editing the brain connectome or the brain would look like is some robot doing this. But the truth is we kind of have some variation on this now that leverages long-term plasticity. And what got me interested in connectomics was I realized that ultimately we as neuroscience doctors had a very large gap in what we were actually able to explain on an individual patient level and what was actually possible to explain. And so we ended up giving hand-waving answers like, oh, well, this is, there's lots of different responses to medicine. And we didn't even have objective measures to know how to know that a medicine was actually hitting the target we thought it was before we wrote off and said, this isn't going to work. And ignorance for doctors, as I think Chris's historical vignette shows, has profound consequences in how we think about the problems we treat and the actions that we take based on that. So I focused a lot of my last many years after becoming aware of these kind of innovations that were running in parallel to the clinical world, of figuring out how to translate better information into the clinical setting. So connectomics is a big data approach, just like genomics. And as Chris pointed out, it's mostly MRI based. And ultimately what it comes down to is most MRI scanners in the world, as long as you're running on something in the last 15 years, which as a developed country, South Africa almost certainly has the majority of their scanners at that level, if not all of them. And what it is about getting more information out of the data to make sense of how the brain's wired. Now, Fmri and Dti are not new. These go back to the 90s, and there's over 550,000 papers in the medical literature on this. So while you could quibble about this or that, specificity or sensitivity, ultimately, the bottom line is that it's fairly, self-evident that there are a signal inside of these scans that tell us about how the brain's working in some form. You could argue what the limits of that knowledge are, but that they are useful is not in doubt. And so one thing you have to think about is, why aren't we doing this on every single patient that we see? Why do we get a scan that's based in the 1970s and, which is just a structural T1 brain MRI, that's been around for a long time. Someone won a Nobel Prize for doing this many years ago now. So the question is, why aren't we basically using this? And there's a real simple chain of events that's led to this problem. And I think everyone on here who's ever thought about getting an fMRI knows what these issues are. You have to go get someone to do it. They got to go into the scanner. They've got to process all this. Usually this has to have a huge knowledge base, and becomes this very niche, difficult thing to get. And when it's difficult, you don't do it very often, if at all. Most places that means at all. And if you're at Harvard, you do it on Christmas and Easter, so only people who've got a month to get ready for a brain surgery, for epilepsy, and not the majority of people you treat. Not the kind of people that Chris is showing you, where these are patients who just go off to rehab and struggle and don't really get appropriate adjuvant treatment. And so because we don't do this very often, the clinicians don't know what to do with the information. Because we don't know the information, we don't demand that we get this at our hospitals, because what are we going to do with it information? And then because we can't get it, we don't demand it, we can't get it, and it becomes a vicious cycle. And so the question is, given that you can now map the brain to this level of detail, why aren't we demanding this? Why aren't we basically saying, I want this on every single patient? And well, it's simple. If I've spent a good portion of my adult life staring at images like this and trying to make sense of them. And it basically is an extremely high dimensional problem. So if you look at this, it's pretty clear that it's hard to know what all these things are doing. And so showing a pretty picture of the brain, if someone's fairly knowledgeable, they can find the arcuate fasciculus and say that's been disrupted, and that's why you have language problems. But this is a really complicated problem, and making a pretty picture like this and giving it to a clinician, even if they're an expert in this area, and especially if they're not an expert, which probably most people on here haven't published 45 papers on this topic in the last couple of years, it doesn't provide us clinically usable information. So let's just go into what an fMRI actually is, and we'll dive into what the real root cause of the problem, which Chris briefly touched on. And then we'll talk about why, to really bridge this gap, machine learning is the only path forward. It's the only path forward in neuroscience. I think that's pretty self-evident. And the simple reason is this: if the brain was simple to understand, we'd be too simple to understand the brain. We would be C. elegans or something. And so ultimately, because of the complexity necessary to do the kind of things the humans do, we need to basically take something smarter than a human, or at least better able to parallel process things, to crack the code. And so if you look at the fMRI, this is the raw image. This is what it looks like. And it's hard to argue that this is better than any of the anatomic images from the 1970 period that have just been cleaned up and improved over the years. It's noisy. And ultimately, what you can do with this is that you do this image multiple times, and you extract how much iron is in every voxel, and that's basically the oxygenation status of iron. Tells you how much you can back calculate how much blood flow is going through, and from that you can calculate brain activity. So what you're seeing at that little voxel in the crosshair is that signal below, which is the bold signal, the blood oxygen level dependent signal. It's the raw signal of fMRI. Now that's pretty cool that you can do that, but the challenge with it is, again, you have that signal happening at all the voxels on that scan. So what the problem with it is that who's talking to who at what time? And this is where you get into this issue that this is an extremely high dimensional problem, just like the DTI showed. And if I just gave you this raw data, what would you do with this? You wouldn't say, well, okay, you need to take an antidepressant, that will work great. But in there's probably the information that we can get out of that actually might tell us that. And one of the things about machine learning that is absolutely a rock solid, truth is that when you deal with millions and million dimensional problem, one of the things that's very helpful to make the models work is to cut down the number of dimensions to something manageable. It's called dimensionality reduction of the data. And what you can see here is the best method that we know for reducing dimensionality, which is parcellating the brain. Divide the brain into regions. And it has a huge advantage to it, because unlike just doing this with raw statistical data where you just have to tell the machine, go find something, it's less noisy and it's more meaningful. So again, we know something about Broca's area, and it's good to look at Broca's area separate from the motor cortex. We don't want the machine to maybe classify those differently because of just raw noise. And this cuts the number of dimensions down from tens of millions to 379. That's pretty good. Now, Chris talked about the Glasser Atlas. We thought about this again as a really big step forward towards being able to talk about the brain the way it really is and communicate results. So theoretically, if I say 8C, we can all talk about it, and we're talking about the same part of the brain, not just the middle frontal gyrus, which the middle frontal gyrus is a construct by humans trying to make sense of the basic similarities of patterns of the soul side gyri. And this is back in the period of time when we didn't have any ability to do something like this. So it was the best we could do in the 1800s and 1930s. But the truth of the matter is the brain doesn't work that way. It's not how it's organized. We know that now. And now we have a way to communicate where it's a little bit better of a map. So this is more based on biological reality. One of the things that the next step of this, and really the big translational step, is it's all cute and well that we can get really great maps of the brain and learn about them. But it's not realistic to ask clinicians or even expect even researchers who are doing that map, because again, I've talked to Matt Glasser and he doesn't have all these things just memorized off the top of his head of what they're doing and all this. And it's just because it's a massive amount of data. So we have to be able to apply this. And the biggest challenge is, how do we take a brain and map all those areas? Well, you can register an atlas, which is a list of where things are in the brain. So Broca's area has a place, et cetera. But when you get into the kind of patients we were seeing in our clinic, that the software crashes, and even here, it's not doing a phenomenal job. It's missing a few sulci, but you can work around that a little bit. But still, this is actually a fairly challenging issue. So what we did to go at this problem is we said, let's do it a little differently. Let's not try to spend time warping an atlas, which may or may not be that patient's anatomy. Let's go to the tractography, the connections of the brain, the white matter connections, and use machine learning to find the best fit. So for example, if you look at this area here, which is Broca's area, we taught the machine how to recognize whether somebody is part of Broca's area in the brain based on who it's connected to. So this voxel there gets assigned yes, and the next one gets assigned no. And it goes around doing that for every parcellation and every voxel in the brain. So everyone gets assigned. Now, so for example, it can adjust the boundary by saying, well, this was assigned initially here, but it's actually a better fit for Broca's area, and this is our new boundary. It does that on average in a normal person about 3% of the time, but where it really excels is when there are holes in the brain or tumors. And it allows us to basically say, well, here are the, given that there's a hole in the brain, or there's a necrotic region of brain tumor, or whatever stroke, you can go on and on. There would be no track, so there shouldn't be a functional brain region in there. And it's smart about doing that. And so, for example, in this case here, this is a patient who had the worst DTI I could find. It's a tumor you can see on the right side of this upper left scan. The frontal lobe's been removed for cancer. This is a recurrent tumor. We didn't reoperate on this patient, even as it was so bad. But one of my residents at the time obtained a DTI scan, so we processed it. And even though the insula is shifted anteriorly, the machine learning finds the basal ganglia. It also knows what's missing. And one of the things, if we start thinking about, there's a lot of applications of once you can map a brain scheme to this level of granularity. And again, if you look at this, there's a kind of a pathway we look at of the simplest one is when we do brain surgery, how do we leave people where they're not cognitively hurt? And we could now map those brain networks with unprecedented detail, as Chris showed. It's fairly simple. We've studied these parcellations. We know who's who. So once we can map it in patient A, we know who the Salience Network is, for example. But it also leads us to much more sophisticated downstream ideas, with the goal that if we understand the connectome, we're better able, and we have a tool that can actually start to change the connectome with essentially minimal or no risk. I mean, TMS is the safest neurologic treatment we know. We could then basically open up a whole new field where what we're talking about is basically rewiring patients in ways that there are some limits, but the point of it is we now have begun to take control of the patient's therapy and rehab in a way that we weren't able to do 10 years ago. So a simple benefit to this is when we look at a tumor like this. So this is a butterfly glioma. It's crossing the corpus callosum. And we've known for decades this is bad. In fact, in most places in the world, this would get a term of inoperable, or someone would remove some of it. But the idea of a complete resection was thought to be too dangerous, and it never made a lot of sense to me, because these things are crossing the corpus callosum. Neurosurgeons have cut the corpus callosum since the 1950s. We know that it has some consequences, but it's not devastating. And this wasn't aligned with the results of going in there and taking this out surgically. Well, now we kind of know why. So one is that you have networks in the language system and the motor system this tumor is burrowing into. But more importantly, when you look at the DMN, which is the most important cognitive network in the human brain, this thing is doing a 270-degree turn around it. So if you went in there and you didn't know that this was the relationship of the tumor to the DMN, you're likely to destroy it, because not like there's a perforated dotted line that tells you where to stop. And so now we actually start to understand what we're seeing. And the most important tool that a doctor has is not a stethoscope, it's your mental image of what the problem is. And so what we started to say was that, given that we can now shine a light in the vast ignorance of the quote unquote association areas, which is an incredibly stupid term now that we understand things, which the association areas are, well, we don't know what's happening, there's something complicated. So we can now begin to think about the brain in a purely anatomic way. And ultimately, physiology plus anatomy equals medicine. If you're not doing anatomy, you're basically doing research. And so what we just, as Chris pointed out, when you talk about how many networks are there, there's not a lot of resources to answer the question. So I went out and found that there's all sorts of network things. And ultimately, the reason, when you look at the fact that there are, a language network and a ventral attention network, and go on and on, one of the questions was, well, how many are there? And the truth of it is that many times, if you take the 7 main networks and you set a more stringent statistical threshold, they'll break apart. So the DMN has 4 major networks inside of it, one of which is the language system. But ultimately, at the most global, the brain, it runs on 7 main axes, and these are what we call the 7 main networks. So to run you through the anatomy so that you're familiar with this, you have the sensory motor system. Interestingly, notice that the auditory cortex is affiliated with the motor cortex, so it's actually closer to that than it is to anything else. You have the visual system, that's probably familiar to people. And then the limbic system. And notice that the limbic system has areas of the temporal pole and the orbitofrontal cortex. So it isn't just the hippocampus and amygdala. Now, if you look at the networks that are less familiar, the central executive network in orange, the default mode network in red, the Salience Network in pink, and the dorsal attention network in green. Those are the 7 big ones. And we call them networks because they fire together. Now, oftentimes they fire together because they're doing a lot of common tasks. Some tasks are completely inside of a network, but many functions come with combinations of these networks that affiliate for short periods of time. So when we say a network, they're not firing together 24/7. They're firing together more often than they fire with anybody else. And what we know is that pattern can be pulled out of resting state data with sophisticated statistical tools and figured out. And one of the things that the first step that we said is there's a massive amount of fMRI literature. But one of the problems is that with the software, it gives you these blobs, and this is not something you can really learn. We need to get it into our nomenclature. So we've invested years now publishing dozens of papers on how do we take the existing knowledge base, utilize statistical tools to figure out what is this in the HCP scheme, and then construct this in networks. Now, this could be days and days. I can give lectures on this for, we have thousands of diagrams and images of this. But I found that in lieu of that, I usually try to get most people on what I think are the 2 biggest updates to what we're doing, that I think that if we're treating patients with neurological diseases or really anything with the brain, that you need to be aware of, because these are things that are relatively non-controversial in neuroscience, but have never really jumped over to clinical neuroscience. And one of which is the control networks. Now, what do we mean by the control networks? Well, they're the networks that's job it is to define the function of other networks. And the DMN and CN sit at the top of it. Now, remember, I told you that these networks can break in half. So what we're talking about is the most essential core, that if we basically say, we tell the machine learning, instead of make 7 networks, make 60 networks. These still stay together. They don't fracture. Other things say, well, the language system is closer to other parts of the language system than it is to the core DMN. When you're left, you have the core DMN. And so what you're looking at is, you think about the brain and how it allocates resources, and the control networks are the parts of the brain that kind of do that. And so essentially the DMN is an internal mental world. So these are things like imagination or recalling episodic memories, that kind of thing. And the central executive network is what you utilize when you're doing something actively. A third and very critical network is the Salience Network. Now note that the DMN was first even hypothesized in 2001 and wasn't really agreed upon that it exists until 2003. The Salience Network is around 2008. So these are not, very old ideas. But what the key thing about it is that they've revolutionized the thinking of the brain, because they clearly exist, they're clearly repeatable, and they play roles in numerous psychopathologies and other disease states. Now, one is the default mode network. You can see this is the core. So there are other areas in that diagram that I showed earlier that are not part of this core. This is the core. It's an anterior cingulate, posterior cingulate, and lateral parietal network. It looks like this. Now, notice that the DMN eventually loops around, and they're parts of the extended default mode network that are, functionally connected to the hippocampus. So it does play a role in memory and things like this, but also it is almost impossible to have Alzheimer's without it have a normal DMN. So it actually doesn't track the disease very well, because everyone has a problem with it. So if you show up in a memory clinic, you have some kind of DMN problem. And this is why it's such an important part of the brain. The Salience Network is a middle cingulate and anterior insula network that looks something like this. And the central executive network is kind of where we think about executive function. So the frontal pole, you have this area 8C, which is in the middle that I pointed out earlier, and PFM, which is in the back inferior parietal lobe. Now, when you can look at the vast literature on these, of what these been linked to, this is covering a lot of ground in the cognitive space. So, for example, most mental illnesses are fundamentally at their core have some, if not many, problems with how these networks talk to each other. But also, we think about the response to pain and emotional regulation. This is a Salience Network problem. So depression has a very serious role linkage to the parts of the Salience Network. The central executive network is involved in working memory and executive function. It's also the main network that develops between the ages of 10 and 17. And it's thought to be that what it's doing is to ultimately help the brain get into very difficult configurations to do deep thinking or align things that normally don't talk to each other very often. And the dorsal attention network is kind of a misnomer, in my opinion, because while sustained directed attention is what it's doing, its clinical significance is as an IQ network. So if you damage the DAN, your IQ drops. Very interesting. Now, where this becomes powerful is when we start to make inferences about clinical observations that we've never understood. And so, for example, if we look at how on the left, this is the medial frontal lobe, and we know that injury to the medial frontal lobe will cause akinetic mutism and abulation. But the problem is, when we say the injury to bilateral medial frontal lobes, we know that that's bifrontal, which is, a fate worse than death in most people. So the question is, what exactly are we trying to not injure? And we didn't really know. We didn't know what the anatomy was. But if you realize that the default mode network, Salience Network, and the supplementary motor area, or SMA, form a little chain in the medial frontal lobe, and when you start to compare this to data from different groups, particularly this group from Harvard and PNAS, and what they were showing was, if you look on the lower left, those areas in red are the areas that if you injure them, you become an akinetic mute. And basically it's the thalamus and upper brainstem and this little axis. So now we have a clear understanding of this initiation system, and the injury to the initiation axis, or what someone at a recent meeting when we were talking about this coined the initiation axis syndrome, is a problem with these networks and how they get you out of your internal mental world and off doing something. Now, ultimately, we talked about the broken Wernicke model, and it's not that this is wrong, because we know that language is basically in these two parts of the brain. It's that this isn't really how, it really doesn't show you everything necessary. And it's honestly, we need to have a real thought with ourselves. Do we think that two guys working in the 1870s, before we even had actually even silver stained a neuron, nailed the language system in such a way that it could not be improved on? And of course, that's obviously ridiculous. So how do we upgrade this? And one of the first things that really took down the house on this problem, and where we realized that this model is not going to hack it, was when people started studying dementia, and we started studying frontotemporal dementia, and specifically a problem called semantic dementia. And this is where you have fluent speech, but an absence of content. Meaning that if I asked you to sound fluent, but if I asked you, do pigs drive cars, you might say, well, maybe if they needed to. And one of the things about that is that the lesion is in the left temporal pole. So it's nowhere near any of these areas, not particularly integrated into these areas. And that really forced everyone to separate out the concept of speech, which is the movement of your mouth, tongue, and larynx and pharynx to make noises that make some kind of sense, and language, which is the cognitive control of that system, into different streams. And the idea that we needed to incorporate things outside of broken Wernicke's area. And so this is the model we have of the speech type pathways. And you can see that there's a lot of areas that are not in the canonical model. Notice that that little guy in the middle, in the around 9 o'clock, is 44, which is Broca's area. So we're way outside of that in some of these. And again, some of these areas are doing predominantly speech, and some of them do motor, or language. One of the things that has come out of the Connectome project, so this is really the last 2 to 3 years, is this area 55B, which is not in the middle frontal gyrus. It's not directly connected to Broca's area, and it's a motor cortex for the larynx. There's now a study recently, in the last couple of weeks, out of Spain, showing that replicating some results we have, that if you injure this in surgery, patients basically can't phonate. So it's a specific problem with laryngeal function. And this is not in the Broca model. This is a patient that we learned this the hard way on. Years later, we couldn't figure out what had happened to this patient. We could write a detailed email describing giving financial advice as a financial advisor, but couldn't talk because he couldn't use his larynx. Well, now we have an understanding of that. And it shows why we really need to use data to understand our problems. So also when we start talking about where does the temporal pole come into this? And we've removed the temporal pole for years as surgeons as a common treatment for epilepsy, but we know that there are cognitive problems. And people just kind of assume that this was the hippocampus, and if you're doing surgery for a hippocampal epilepsy, this is just the price you have to pay. But the reality is that's not really the whole story, because when we developed laser therapies that you could basically stick a laser fiber into the hippocampus and burn it without cutting into the temporal lobe, we didn't see this problem as much. And so we realized the hippocampus wasn't everything there. And so you see this thing in yellow. This is every area that's functionally firing at the same time as the temporal pole. And what you can see is that not only is there this little stream of things that from the Wernicke areas to the temporal pole, which is covering sort of a language processing stream. And, obviously the temporal pole is talking Broca's area. You can see that there, but also in yellow, those areas are the default mode network core. So language is part of the default mode network. And so the DMN plays a role in the production of language, which isn't something we were taught in medical school. So we look at this, all the things, and there's some other things that, those last two images could be a different part of the talk, but just in the interest of simplicity, this is actually what the real model is closer to. And again, this could get more complex, but the bottom line is without connectomics, without mapping the brain, you really can't figure this out. And bad things will happen to people you do surgery on because you just don't know where all these areas are. And we just accept them as reality when they may not necessarily be unavoidable in some patients. Finally, I want to show you something that is utterly fascinating. So if you look at this, we have those 7 networks. And of course, if you look at the CN, DMN, Salience, and DAN, They're kind of just blobs over the brain. The other ones make sense. They're kind of localized in one or a couple of areas. But the C and DMN are more complicated, more diffuse. And so it looks, when I looked at this, I thought, well, you just have to memorize this. But there's actually a real logic to it. So first look at the interhemispheric portion. And that's not an accident. That's patterning. And we see this all over embryology. And if you look at the DMN, anterior, posterior cingulate, anterior cingulate, comes on the hemisphere, down to the operculum, back to the parietal lobe, down to the temporal lobe, it makes a sort of S. But sort of the CEN does this too. Salience network does something like this. And the dorsal tension network, which is older in evolution, which hugs the deeper structures a little bit more, looks something like that. So this repetitive pattern has been tracked all the way in evolution to at least marmosets. Probably goes a lot older than that. So this is not an accident. This is kind of a segmentation approach of how these areas probably form together in some way and then are dragged through the cortex over time as the brain folds. Now, if we look at the cerebellum, it turns out the majority of the cerebellum is not motor. Okay, the blue on this diagram is motor, but the majority of it is actually expressing the control networks. And when you look at this in the parasagittal cut, if we look at started about 1 o'clock on the dial on this here diagram on the left, and you go counterclockwise, motor, dorsal tension, salience, CEN, DMN, and then it reverses and does that in the opposite order. So it's the organizing principle of the cerebellum is the control networks. That's how it works. And this is the same true in the basal ganglia. So the control network organization is one of the fundamental organizing principles of the human brain. Now, again, if you look at the pipeline that we're doing, we take a standard generic atlas. We use machine learning, as I talked before, to make it personalized, which can handle distortions or individual variability. We then use this to slice and dice the data up, where we look at the functional connectivity between every area and every other area to make a large grid of connectivity, and then we use other tools to make it make sense. And so I want to kind of dive into what functional connectivity actually is, because most clinicians have never heard of this or only briefly. So as Chris pointed out, when you use a task, areas are activated that are relevant to the task. But it turns out the same areas are constantly activating each other in different combinations. And this is going on all the time, whether or not you're using it. So when we go to the task activity, we're not going from 0 to 60, we're going from 40 to 60. And so what you can do is you can study this, and there are patterns. So some things are supposed to fire together, other things are not supposed to fire together. And in a healthy brain, the correlation, meaning how the frequency of firing together, should be relatively consistent. So there's maybe one person's thinking about A or another person's thinking about B, and different areas fire together, but there's things that aren't supposed to happen. And so using machine learning, you can start to highlight them as outliers. And we don't know for sure that an area that has 20 outliers with another area is working abnormally, but it seems like a good place to start. And Chris talked about this paper, so I won't belabor the point, but it's clear that resting state connectivity is disease defining. You can actually look at this, and when diseases have different symptoms and different responses to treatment, even if they're similar, we call them different diseases. So there really is no such thing as depression, schizophrenia. It's all circuit problems, and the circuits define the problem. Now, again, when we look at this, this is the data set that our software puts out. It's a massively high dimensional, depending on how you define it. It could be up to 25 million dimensions. But what you're basically looking at is red areas or areas that are highly correlated, meaning that they're firing together a lot, and blue are not anti-correlated, meaning they're not firing together. Now, one of the nice things about it is we've invested a ton of effort into figuring out who these guys are. So we kind of can make some inferences. And when you utilize powerful statistical tools and prior knowledge, resting state fMRI has a huge advantage over task, because you're getting all the networks, all the functions at once. They're all in there at some point. They're all firing together at some point. And more importantly, you could do this on anybody. It's just like laying in an MRI. So there's no complex PhD level task to do this. You just lie in the MRI and you get the test, and then we use the statistics to make sense of it. And so what we want to do with this is, if resting state's easy, let's optimize to make it as effective as possible and to make it so that eventually the goal is that it just gets put straight from the scanner into the cloud. You get a full connectome, and so it's like a sodium test. So then there's really just no reason to not do it on pretty much anybody, because you can get it all the time. Why not? And that was our goal with constructing this. And ultimately, what the longer term goal is to make sense of data like this, you have to know facts about the patient, like what were their symptoms, what did they respond to treatment. And there's a lot of data suggesting that if you use machine learning, putting into a cloud architecture and leveraging these kind of tools, you can begin to make sense of this. And so if you think about the fact that we can now map the brain, and we can do something about it in many cases. We can change how the brain works. There's a real key link that I think we all have to make in our head, which is that every event, every emotion, whatever, even whether it's caused by neurochemicals or ill humors or whatever you want to think people come up with to explain mental illness over the years, at the fundamental level, there's a circuit that's malfiring somewhere in the brain. And that symptom circuit is knowable. And so this gets into the idea of the transdiagnostic hypothesis, which says that if you have anxiety, whether or not you got there through schizophrenia, depression, or whatever, there's a circuit, and it should be similar sets of circuits that are problematic regardless of the diagnosis. The diagnosis is relevant. So if you have 3 symptoms, you have 3 circuit problems, and these are 3 simultaneous issues. They may be overlap between them, but they're knowable. And that's a revolutionary idea. It takes psychiatry and turns it into almost surgery. And it's really, to be honest, it's about time we provide the psychiatrist with adequate tools, as they're the only doctors who don't look at or image their organ on a regular basis. Everyone else orders images all the time. Radiology's been around for a long time. But there really is no reason to do this until we have the tools to actually adequately image the disease. And it's critical because mental illness is just really an epic problem. So you can see here, this is a diagram of schizophrenic symptoms, and all the overlaps are how many times patients have overlaps between the symptoms in a large cohort of people. And this is messy. And even if you get to the level of trying to sort this out, these patients are all a little bit different. And we all kind of know this. So when we use an RCT, we're basically just saying, well, if you didn't know anything other than the patient kind of has schizophrenic type symptoms, it's good to start this medicine. That's not really the platonic ideal of how to treat patients. The closer to the platonic ideal is that for the 30 symptoms of schizophrenia, we map them to circuits and we do something about it. It may not be TMS, it may be something else, but we do something about it. And we can't do something about it if we don't understand it. So this is a really complex machine learning tool we put together. We have lots of these kind of algorithms that are aimed directly at answering clinical questions that doctors care about. And what our goal is to map symptoms to circuits. So I'll touch on it a little bit. This is a project in schizophrenia we did with a group in China. And what we looked at is we said, okay, let's take all the symptoms of schizophrenia, map them to circuits, and disentangle them from each other. So on the left are the areas of the brain that the machine learning tells us are important for telling whether a patient has hallucinations or not. And you can see on the right, it's telling us that it's the auditory and visual system. Makes sense. Good to know. But we start getting some stuff that we didn't know. So when we talk about abstract thought, what was shocking to us is that what drives abstract thought is the visual system. And when we look at this, actually a substantial amount of the white matter of the brain is linking the visual system to some other part of the brain, usually the frontal lobe. So why would the frontal lobe and temporal pole need to talk to the visual system? And the answer is because that's how the brain works. It tells the visual system what to process based on goals. And when you have problems in those areas, you can't think straight. But we didn't know that. Machine learning found that out for us. And again, I'll point out one last really interesting one. When you talk about people who have somatic delusions, people who have somatization, this little area in the insula shows up high on the list. And that's an area of the insula that's actually synchronized with the stomach rhythm. They fire on a common unit. So it's really fascinating that a functional connectivity problem in that area causes people to think that they have problems in their abdomen. So when you think about how do we leverage this kind of insight and do something about it, we talked a lot about TMS, and the question is, how do we utilize an active interventional and rehabilitation program? Meaning we know we figure out what the best idea of what we think is probably wrong, or at least the best guess we can, and we do something about it. And all the tools exist for this. And if you look at TMS, essentially it's inducing a field in the brain. Now, it's not a focal field. You see that there's green that kind of crosses even to the other hemisphere. And so for a long time, people thought that maybe you didn't need to be very specific. The data is pretty clear now that you need to be over the correct area. You need to be, just even though a lot of areas are getting electrical stim, the stuff in the red area is really what's getting the benefit. And so when we look at these connectivity matrices, because we know who the parcellations are, we can start to make some inferences on it. So you look here, this is a subset of that big matrix I showed you. And all the guys here on the X and Y axis are areas that are involved in language function, okay? We know that because you saw the model that we had. So it's mostly red. And it's mostly red because the language areas fire together, even when you're not talking. On the right is a patient with a glioblastoma in the arcuate fasciculus, and you can see that it's mostly blue because those areas are not talking anymore. So it's pretty cool that we can actually see aphasia. But beyond just an intellectual interest, you can actually do something about it. So that column is at area 55B, right? So it's a place in the brain. So if we know that this is how the connectome is working in this patient, we can actually make that into an object, export it, and do something about it. And to show you that this is not a placebo, in case Chris's presentation didn't provide some convincing evidence, this is a patient with a fairly devastating left MCA stroke. She's 18 years old. And after a year or so after the stroke, she's really had some improvement, but significant problems with communication, which you shouldn't be surprised. So chronic deficit, usually at a year, we kind of think that these patients generally don't improve. And so we looked at her through this, and I'll just spare you the whole details, but basically we treated at that arrow. And the reason we did is we looked at the damage and saw that the supplementary motor area and the initiation system didn't talk to the language system there to destroy it. So we thought, well, let's try to get the other side to take over. So we stimulated there, did speech therapy. And you're looking at this in a coronal view, so from the back. So on the left is the language system. And on the right is 4 months later, after her language had made significant improvement. And, you know, that's quite a bit of, I mean, I don't think I have to go through and interpret that there's now this thing grew connections to the other side of the brain. And we repeated this enough times to know this is not just an imaging artifact, this is legit. So again, very much not a placebo. Now, again, when you look at this on the left, the raw data is blue and red. And the problem with it is we can create these great montages of looking at how different networks talk to each other. In this case, it's the DMN and CN. But the real benefit is when we start to be able to look at it and say what's not normal. So this is a machine learning algorithm that basically just, again, as Chris says, flags outliers. Now, some of the black is blacked out for a reason. It's because some areas are noisy, and that's just because one person maybe thinking about soccer and the other person's thinking about World War II, and these activate different areas of the brain. And we don't want to call normal cognition noise or disease. We just black it out. We say, don't know what it is, we're not going to show it as a pathology. But there's certain things that are never supposed to fire together or always supposed to fire together. And this is the fundamental basis of mental illness. So while you might not get the exact same resting state on the same person twice, if someone's depressed, we expect that there's something wrong with the core working of how the brain is running its default state that we're drilling down on. So how do we do this? And I'm sure Chris is going to go into this in future lectures, but how do you actually go and pick targets? And the answer is, I had to figure this out because I was the first person to have the software to mess around with this. And so the basic way to think about it is to go back and think like a doctor. Think as if you were basically dealing with a broken arm or cold or pneumonia or COVID-19 or whatever. You go and look at it and say, what do I think is wrong? What is the status of this? What is the anatomy? So it's a case of a pneumonia, you would get a chest x-ray, say, well, what do the lobes look like? What's the extent of the disease? What do I think is happening? And then come up with a reasonable plan that addresses these issues. So what's most important is to get a good understanding of the chief complaint. What are you here to actually fix? It might be multiple things. Many times brain injury damages multiple systems, since people have aphasia and hemiplegia with a stroke. So we have to look at that and say, okay, well, maybe we have to put a fixated thing and say we can only tackle 2 problems right now. Sometimes multiple, treating an anxiety also hits something else because they're working on the same set of circuits. But the bottom line is to sit there and define what the relationship we're trying to do. What are we trying to make better? How bad is this? And what's the realistic expectation? So if someone's neurologically devastated, we might be happy if they get anything back, and it's, 5% improvement is hard to poo-poo when it's your 5% neurologic function. And we know this from spinal cord injury that even doing a surgery that reduces the injury from C5 down to C6, we save 1 level, is the difference between having your bicep in order able to feed yourself or not, which is a pretty substantial thing if you're, essentially a quadriplegic. And so these little things make a huge difference. So actually the people who get the most improvement in quality of life are not the people we make normal, it's the very sick people that we make slightly better. So the first step is to go and look at the structural images. And again, this goes into going into DTI. Now, in some cases for mental illness patients, you can kind of just quickly skip this step. So unless there's a tumor or some major thing, we're not going to see much on the gross structural images of a mental illness patient that's going to inform us. But for people who've had strokes and other things, it's critical to know where the hole is. Because, for example, if the corticospinal tract has been injured, well, they may not, if it's cut, you're probably not going to fix this with TMS. You're not going to it's not going to grow across the gap. But that's really rarely completely severed. And so sometimes you actually see patients that are functionally normal, but structurally damaged. So a good example would be, well, there's, instead of having millions of fibers to the spinal cord from the motor cortex, you have 20. Those 20 work fine, but you only have 20. Well, that's going to be weakness. So the thought process there is, what can you do to be helpful? The Hippocratic Oath. And what you could do to be helpful is try to make those 20 work better. And that may be the best you can do. Whereas what we find a lot of times is there are patients whose weakness is actually the motor planning systems or the control systems around the motor cortex. And it's a functional problem. So that's a very different situation where your goal there is to realign the functional connectivity to make them work better. And so tools now that you can tell, well, something is missing or damaged is a big step forward. And once we take that into consideration, we think, well, what's the primary mechanism of disease? Is this a hole in the brain, such as a tumor or a stroke or things like this? Is this a diffuse problem, such as trying to improve Parkinson's or Alzheimer's? Or is this basically a rhythm issue where we don't think the brain cells are dying, they're just the thalamic cortical rhythms are off in such a way that areas are firing out of sync or in sync in ways they're not supposed to do. And so once you have an idea about what you're there to do, what's the nature of the structural problem, if any, we have an idea of how to put together a montage. And a montage is, what functional connectivity comparison do you want to do? And so again, functional connectivity, what we're doing is we're looking at the bold signal and comparing it everybody with everybody else. So it's all combinations. And in this case, you could compare DMN and CEN and look and see how these two networks, all the parts are talking, and pick a reasonable strategy. So actually, let me zoom ahead here real quick in the interest of time. And so there are different montages that are useful. So for example, if we're dealing with mental illness, looking at the default mode network and CEN, of how they talk to each other or how the Salience talks with other ones. These are ones that are very useful because this is really where the mental illnesses happen. Whereas if someone has a language disturbance, you look at the language system or how the language system talks to the DMN. And there's, you can get creative and keep going, but there's some common ones that people learn really quickly that are workhorses. So DN CMN is almost a running joke in our clinic because I hit it all the time. It's one of the first comparisons we do for a lot of problems. And so usually we try to aim for about 2 targets. The main goal is to, ideally, if it can be at 2 sides of the brain, that's nice, or 2 different lobes. We don't like to hit 2 things next to each other. But a lot of times you try to preference it. So if something shows up in the dorsolateral prefrontal cortex, the frontal lobe, well, we know that that tends to work pretty well for depression. So if area 46 or area 8AV or some common targets we see are abnormal, we don't go looking for other targets. That generally shows up on the list. You say, well, we know that works a lot of the time. But not everyone with depression has abnormalities in that target. And again, we talked about this. And so the point of it is, there are different reasons to do different things, of which sometimes it's helpful to look at the raw data. But the bottom line is we found the anomaly detector is more easy to work with. And as Chris mentioned, ITBS increases the area and CTBS turns it down. And so interestingly, you can also pick some other interesting targets like cerebellum, some areas in the anterior temporal lobe. But if these are the muscle, you might not be able to do something as aggressive as theta burst. Theta burst is beneficial because it's faster, but some of these other protocols they may take longer, but they're more easy to do in some of the more painful areas where the muscle might be stimulated by the activation. And finally, we think TMS works by setting the brain to a state by which neuroplasticity is more possible. So the way I describe it is if you had a malaligned jaw, the way the dentist fixes is they break your jaw and they reset the bone. Well, this is the brain's version of that. And so we think it's really important that we combine this with therapy. So it isn't that this is a replacement for therapy. This is to get as much out of therapy as possible because we think it's really important. So I'll show you a few cases. So with sensory motor cases, again, really important that we know the structural architecture and that the sensory motor system is motor in the motor cortex. There are areas that do planning and sensation things that are important and not working normally. And so you can look at the fact that there are even things in the cingulate gyrus that are unique motor areas. And again, you can look and see that the motor system has an extensive connections to the rest of the brain, not just the corticospinal tract, but connections to the basal ganglia, and connections to each other. So when you have an injury to one side, sometimes the other side becomes overactive because they're normally keeping each other in balance. So if you look at this case, the motor planning in this case is different because there's a hole in the motor cortex. This has been there for 8 years. So if you have to have realistic goals, there's corticospinal tract, but we're probably not going to make our arm normal. We looked at that though and said, well, the leg seems okay. Maybe we can do this. And this patient had an almost plegic leg, was working on spastically. And so because she didn't have a significant functional connectivity problem, we looked at this and said, well, we're going to aim for the medial cortex with the goal of trying to get as much as we could out of that. And so we treated mainly based on the structural data. In this case, this is a similar problem where a brain tumor was taken out of around the motor cortex with paraplegia. And as you can see here, what you're seeing is that not only is there decreased connectivity on one side, on the good side, the bad side is overactive. So we came up with a strategy to turn down the good side, the opposite side, with CTBS and turn up the bad side. And I'll save you the video, but bottom line is Chris can testify that the patient made a pretty dramatic recovery. And again, this patient woke up hemiplegic, and we didn't have an explanation for it. Corticospinal tract's fine. So it looks great even. So why is this patient paralyzed? Well, in the past, we would have said, well, I don't know, maybe we cut some tracts or something. But the reality is, no, it's not at all. We cut the cingulate motor areas. There's even connections from the other side. And when we looked at the connectivity, you can see those blue columns. That's not supposed to be there like that. And so we basically, picked several targets off this list where those arrows are to try to turn them up. So this patient didn't have a paralysis from a structural problem. They had a paralysis by removal of an area in the motor planning system that threw the whole system off. Again, with the language system, you can see a much more sophisticated system here. And we talked about that band of structural connectivity. This is the accessory language system. And again, this gets fairly complicated, the type of montages, and this could get, and we could go deep into the weeds, but there are lots of different ways you can combine language with vision to understand reading. You can combine language with DMN to understand the cognitive components of language. You can combine language with motor areas to understand dysarthria. And again, it really just comes down to, we need to be doctors and utilize software like this to think like doctors, to explore different systems. We know a lot about how the brain works intuitively from being clinicians. This just is aimed to make us smarter clinicians. So in this case, this is a patient who had a previous brain surgery, and she had some language disturbance, but her real problem was reading. And if you look here, structurally her language is okay. She's missing the temporal pole, which we know is a cognitive component of language. But why reading? Well, it's simple when we look at here. So if you look at this anomaly detector, the montage that we picked is language in blue, DMN in orange, and then we looked at the medial temporal lobe in red. So we're looking at what she's missing. She's missing the medial parts of the medial temporal lobe. And what you see is that there are numerous ways that the language system and the visual system have numerous interconnected anomalies, because something about what we did to the temporal lobe threw the system off. So we picked those two targets there, and there's language with just vision only. You can see that there's just so many things that are downstream effects of doing that operation. And this managed to improve her reading by about 60% on reading scores. Again, finally for mental illness, which is a common one, we tend to, there's lots of different ways to do this, and I could give a whole lecture on this particular topic, but in short, scans weren't very helpful. But when we have a patient who, in this case, who's failed 2 medicines, has PTSD, we can now begin to look and see that there are areas like area 46, which is a common TMS target. It's actually part of the FDA approved target, and area 8AV, which we didn't even know what that was, but it's basically abnormal in everyone with anxiety that we've seen so far. We now have a clear path on how we can change those and turn those down. So I'll leave it there. That's a lot of information, and if anyone has any questions, I'm happy to take them. Thanks.
Thank you so much, Prof. Sugru and Dr. Profius. If there's any questions, please feel free to either unmute and ask, or you can go ahead and chat them in the chat box.
As far as Tourette's, Tourette's and OCD are really fascinating problems, obviously. And what's interesting is OCD is FDA approved TMS target. So we kind of know what seems to work across the board. And Tourette's is very similar. We know it's a caudate basal ganglia motor problem. Ultimately, at the end of the day, it's a compulsion. And what was really surprising about OCD was they tried to treat the CEN DMN and it didn't work. There's a lot of studies that had negative studies. What came out of it was somebody at Duke, for reasons that are unknown to anybody, ran a trial where they stimulated what was basically the supplementary motor area, and it worked. And that's actually the now approved target. So it turns out that the SMA it picks between what do you want to do, and again, it's the area that starts to know, like we know that it's predicting what you're going to do before you're aware that that you want to do it. And so it makes sense that if that area is getting the wrong signal from the basal ganglia, you could feel compelled that you're about to do something or you need to do something. Now, how this gets into the cognitive distressing components of OCD and things, but for Tourette's, that's fairly simple. Now, the challenge with it is, of course, we don't know whether it's DBS that's the right target, or TMS, or both, or TMS is easier. DBS is probably more permanent, because we can reprogram someone, but they can always slip back into the same thing. And particularly for serious or genetic disorders, it may be unrealistic to just stimulate a part of the brain and hope that it stays that way and changes the whole connectome if every cell in the connectome has the same problem. But the bottom line is, it's not that we have a solution for it. What we have is a tool that on an individual level can actually look at the patient and start to think through this problem. And our goal, if we have enough data, is we would start to know, and maybe we find out that there is no real Tourette's, there's 3 or 4 Tourette's. As far as epilepsy surgery, the thought process to think through is that, of course we know that when we find the target, it's and do something about it, it's life-changing, right? If you get rid of the epilepsy, no question. One of the things about it is, we know that there's two basic problems with epilepsy. It is, number one, finding the target, and number two, that we don't work to get to patients early enough. So the average at UCSF, for example, is the time from failing your third med or second medicine, when we know that you should be getting an epilepsy workup, to actually getting that epilepsy workup is 8 years. So we're not aggressive enough. But the second thing that come out of some of the Mri experience is, if you don't look for the focus, a lot of times you don't think there is a focus. Some people don't, we've underestimated the number of people. And if you put SCG electrodes, which are relatively not that invasive, you can oftentimes find epilepsy where you didn't expect it. But the question is, if you have non-lesional epilepsy, and it's not immediately clear where it's coming from on surface EEG, where do you put these electrodes? And ultimately, with epilepsy, there's an evolving literature of a handful of papers that show that resting state functional connectivity can give you a better idea where the focus likely is. Now, I don't think it's to the point where I would recommend doing an operation based on it, but you can make sure that you're putting your electrodes and covering the abnormal targets. And what that means is there's probably people for whom surgery is available and life-changing that we're underestimating because we don't have any facts or thing, but it's hard to justify just sticking electrodes on all of them. Well, if you have some data, you say, well, let's break the tie in our head with data. And that's a pretty significant change, because given the laser interstitial ablation is such a good treatment for epilepsy, if you can find it to one focus, it's not that big of a deal. It's not just finding these stupid things. As far as functional neurologic disorders, best way to think about it is that there are probably some of these patients have almost all of them have some kind of connectivity problem. So the reason they have lots of mental illnesses is that part of the greater mental illness is a functional connectivity problem. And if the functional connectivity problem is in the emotional areas, you feel sad or act out. And if it goes into the areas that understand pain, like the Salience Network, or the areas that feel your gut, like in the insula, you have these bizarre symptoms and so what happens is a lot of these people have overlaps in this. The answer is, obviously, we know functional neurologic disorders. The frustrating part of it is we can't have any objective evidence to back this up. And so it either goes into people writing them off, or, people trying to be helpful, but, doing things that are not that useful in many cases, or psychotherapy or pseudoscience. And none of those things are the ideal scenario. The ideal scenario is there's something in the brain in these patients that's convincing them that their gut hurts or their back hurts. And we can now answer that and study it. So we don't have all the answers to it, but we have a better way to look at it. As far as the pediatric population, the one challenge is actually just getting the imaging. So if you're looking at functional MRI, if you have to knock the kid out, you actually change the connectivity pattern of the brain. And so, for babies, it's possible to do fMRIs, but it's very challenging. And it may not be something that can be done clinically very often. So giving somebody propofol anesthesia, it's going to affect it. And second thing about it is, of course, we know the connectivity is a moving target in kids. It changes. Having said that, we're working some really interesting data out of Singapore that shows that our algorithm does some very interesting things in kids. And the bottom line is, we think that in the near future, we'll be able to recognize that if certain things are developing in a 7-year-old, that they're likely to be a depressed teenager. And that's starting to become a very plausible thing based on the data, but we haven't got the patterns down yet. But we have some really, really interesting data on it. So not to the point where I put it in a talk, but I'll just say that it may be that what we start doing is thinking about that you are likely to develop this problem, and can we start intervening when you're at 8 and 9 so that you don't have a life of, being either a troubled writer or someone inside mental institutions.
Okay, thank you so much. I think that was the last question. Thank you, both of you, to Prof. Sugru and to Dr. Profius for this incredible presentation. I think it was really just wonderfully insightful. And I think everyone on this presentation definitely learned some incredible things and got a lot of insights. Just as a reminder to everyone, this is a series and there are sessions planned for the coming 7 weeks at the same time. If you haven't registered, you can use the same link that is in the chat box to register for following sessions. And then for today's session, if you'd like to get CPD points, please don't forget to register, just to make sure that we have your MP number and are able to allocate your CPD points appropriately. But other than that, we hope to see you in the following sessions. I don't know if you want to say some closing words, Chris?
Just like to thank everyone for a late evening. It's a completely new way of looking at the brain, and initially it's intimidating. So when I first got exposed looking at the brain at this level, you're almost like a deer in headlight. But I promise you, the more energy you spend at looking at brain disorders in the brain in this way, you will start to get answers or much more relevant questions than what you've ever had. And I'd just like to thank Mike for getting up really early for our talk in South Africa. Thanks, Mike.
And I think that one of the key ones, Chris showed me this the other day, and it's a great case of a patient who had a traumatic brain injury and who basically lost their clicks. That's their only problem. And many years ago, this would have just been a bizarre thing that we would have said, well, I don't know what it is. But we looked at the connectome and found that it was a direct hit to a part of the language system that's not in Broca or Wernicke's area. So I said, that's a uniquely South African neurologic connectomic problem. But the point of it is the fact that you could actually go in there and answer that question for the patient and maybe even do something about that is, you know,
It's actually
a real,
improved quite a bit, Mike.
yeah, yeah. Did you stimulate it?
Yeah.
Oh wow, it's amazing. I told you have to write that up because you're the only. There's only 2 countries in the world where that case report could come out of. All right.
Thank you so much. I don't know if there's any other sort of closing words. Richard, would you like to say something before we close?
Thank you so much. I think that it was a long session, but a worthwhile one. And I think Christos hit the nail on the head there, that it is quite daunting. There's an extraordinary amount to take in here. But I think after 7 weeks, we are all going to feel, those of us who are, you know, complete ignoramuses in this field, are going to feel a hell of a lot more educated. And I'm just thinking about the number of cases I'd like to refer to Christos after this talk, who attended. Thank you so much.
Thanks, everyone.
Okay, thank you, everyone. Please don't forget to register for the following sessions. We look forward to seeing you next week. Thank you. Thanks. And have a lovely evening.
Bye-bye. Thanks.
All right, bye.